All right. Good afternoon, everyone. I hope everyone can hear me well. So, of course, afternoon Central European time. For those of you who are on the East Coast, good morning. And for those on the eastern side of the world, good evening. My name is Kris Modig. I'm leading scientific solutions and enterprise growth at Amass. And today, we're going to focus on how to do scientific work in Claude, going from a general AI to a domain expert. And just to get started, first a bit of background on myself. Most of my professional career, I've been in life sciences and software, working with both. And now at Amass, I'm on the customer-facing side, working mainly with our enterprise customers. And for today's agenda, we'll start with a general introduction on what Amass is, and of course then jumping into Claude, which is also the title of the webinar. So getting started on how to connect Amass using project and skills in Claude. Then we'll walk through some real-world examples, evaluating some assets in the clinics, target prioritization as well. We'll see how much time we have in the end. And also talk a bit about replicable workflows, so how to build these kinds of workflows where you need to run multiple different tasks into one that you can run continuously and more reliably than individual prompts. And then we'll wrap up with a Q&A at the end. So we'll have 45 minutes for the presentation, the demos, the walkthroughs, and then some time for questions as well. So if you have any questions, you can type them into the chat, either at the end or then during the webinar. We'll try to pick them out there after as we go along. All right, so starting just briefly with Amass and what the Amass platform is. What are the cores? So just to give you the background, Amass is basically the, I would call it the intelligence layer for life sciences, or that's what we're trying to build. And you could call it also scientific intelligence infrastructure. And we're built upon the principle of headless intelligence, which means that essentially our core architecture, which you can see outlined here, I'll explain that and what that is in more detail in just a bit, can operate independently of the interface through which you're then using or extracting data information from there. So we have the application as one, we have the MCP, for instance, Claude or OpenAI, and the API, the application programming interface that you can also use then to access and query the data sets that we have across the different cores. Amass can be used by researchers, apps and agents, and developers. So basically these three use cases, these three entry points can then be used by many different kinds of people across organizations in life sciences. And as I mentioned, today we're going to be focusing on the MCP. So that is Claude in this case, but we also have an app in ChatGPT. So that's also something that you could potentially use. And to give a bit more background of the cores, just jumping to the Amass website here, what the cores essentially capture are different things. So we have the DrugCore, GeneCore, PubMed core, trial core, patent core, and regulatory core. And together these form that intelligence layer, as I mentioned. So for instance, in DrugCore, you'll have drugs, of course, so different molecules, and you'll have information about their mechanism of action. You'll have cross links to other cores. So as I'm hovering here, you can see that these are actually linked to, for instance, then targets in the GeneCore. These are linked to literature in the PubMed core. The resources here span from, for instance, PubMed, full text articles to then abstracts and so on. These are again linked to the trial core, also to the DrugCore, so clinical trials, FDA, EMA, and also global trials. And the patent core, so any associated patents that you have to drugs, and then that are also referenced or mentioned in literature, going then back to the regulatory core, so regulatory filings, for instance, from the FDA and EMA. So all of this is linked together, and that's the power of it as well. So you can actually traverse, let's say you need to find a drug, you can traverse that and find relevant literature, find relevant genes, relevant trials, and bring this all together. And that's what we're going to also look at today. So what's the value of Amass? Just in brief, we really try to anchor the any kind of queries you make, any kind of questions you ask, either your favorite LLM provider or in the Amass application, with then concrete references, concrete citations, identifiers, and really have this evidence first mindset. And this is then based on the more than 50 million documents that we have or make available through our course. We also have optimized retrieval, so you can actually find the relevant records. So a lot is happening in the background as well to make sure that you get the right records. So that's also an important part of it. And of course, then providing the accurate identifiers, links, and references in the output as well. So this all leads to a connected knowledge base that you can then gather whenever you're looking for this kind of medical data, for instance, or anything related to papers or patents or trials. So that's it for Amass in brief, and we'll use Amass through the connector, and I'll show you how to set that up in Claude in just a bit. But before we start setting up Claude and jumping into Claude itself, there are a few things that we need to keep in mind, especially when we're working with, well, in life sciences, in research maybe, development. We need to be rigorous, methodologically rigorous. So we cannot just do anything. We need to have the right citations, references. And that's why I went ahead and asked Claude, what are the most important and maybe not obvious things to know when doing scientific work in Claude? And I got a few answers. I collected some of those that are relevant here. So the first thing is results not retrieved. And this means that whenever we're searching for specific knowledge, we only return the top results. Claude only, let's say, makes a web search and returns the top results. It doesn't mean that we look through all of the results, and the ones that are actually omitted from the analysis, they're not considered in the answer that you get either. And that's one thing to keep in mind. That's why it's very important also to get the relevant results back into Claude and have then Claude reason around those. The second one, numbers. So instead of reasoning numbers, should always compute them. So make sure that Claude actually calculates anything numerical in a sandbox using Python, for instance, and not just reason and come up with numbers that look reasonable. And the third one is, and this also ties back to Amass, that we need to anchor claims to actual identifiers. So especially when it comes to this kind of work. So this is what Claude said. I can fabricate a plausible identifier, and that's not what we want, right? So for instance, a PMID or an NCT number that looks right but doesn't really exist can be fabricated by Claude. So we need to make sure that we actually capture the right identifiers and are able to always link back to that and go back and see that this is actually a real piece of information that we're relying on in this analysis, whatever analysis that we do. There's also a knowledge cutoff date in Claude, which means, so the pre-training data set of Claude and any other model for that matter. So we need to use search for anything fresher than whatever that cutoff date is. And that also ties back to the results. So what we don't see, what Claude doesn't see, doesn't necessarily get captured in the analysis. And of course, design is also important. So how we go about designing our queries are a series of tasks in Claude. And this is what Claude said, I'm agreeable, and that's dangerous in hypothesis testing. So we need to make sure that we're not only searching to confirm our hypothesis, we also need to take the other side and let maybe Claude take the other side and argue for the null hypothesis, or then have another agent try to refute the results and get two perspectives on the matter instead. The same prompt doesn't equal the same answer either. So, of course, LLMs are not deterministic. So that's why we need to give structure and guidelines and also the right tools for Claude to work with to also get consistent results. Retrieval provenance, and this basically means that we are able to maybe not only link back to the original sources, but also understand how we got to those sources and be able to replicate that kind of search or query, and potentially also log the steps on how we got there and be able to replicate that afterwards as well. And then finally, verification. So again, this maybe goes back to the design here. So you need to review the claims and sources without necessarily seeing the reasoning, or let an agent do that. So have a fresh pass of whatever task and try to rederive that result without giving the reasoning steps to an agent, or for yourself, for that matter, to look at it and say, oh, that's reasonable, without actually truly verifying how we got there. And now let's get started in Claude then. So in general today, I've prepared two different examples that we're going to walk through in Claude. The first one is a live walkthrough of assessing clinical and commercial asset, and this is using the Amass connector. And the other example is then either a target prioritization or indication landscape analysis, and looking at some of the outputs that we can create in Claude. But let's start inside Claude itself first. So here we go. Here we have Claude. And as you see, I've created a blank webinar project here now. And right now I haven't connected anything into Claude. I have a couple of skills in the background, but also show you how to set one up for you later. And I see there was a question about getting access to slides later. So you also have access to the recording afterwards. So you can always come back to that. So if you miss anything, we'll have the recording ready for you after this as well. Okay, so back to Claude. I assume everyone's more or less familiar with Claude, and actually maybe as part of that we can take a quick poll on what you're actually using internally. So we'll have people vote here. Let's see who wins. So I assume that the majority of people here are using Claude, at least to some extent. We have some ChatGPT, Gemini. So Claude is definitely predominant in this crew. And this is really useful to know. So, of course, anything that we do in Claude can also be carried over to some extent to other tools as well, like ChatGPT or Gemini. And same logic, similar strategies to actually creating prompts or creating skills or instructions can also be applied to some extent. Okay, so now that we're in Claude, I'm just going to start by setting up Amass here. And in order to set up Amass yourself, you do need to have an Amass account. So just make sure that if you don't have one, you can do that afterwards by just going to the Amass webpage. And you can click on try for free here. We have the app or MCP connector that you can just sign up for, and then you'll have access to Amass as well. So in Claude, I'll just go to settings here and connectors. And as you can see here, I already have Amass here. If you don't have Amass, you can browse connectors. So just add and search for Amass. And you can see here we have a verified Amass connector. Click on the plus icon to connect to Claude. And then we need to authenticate. And this just verifies with Amass, that you have an Amass account, have a user account, and then we're basically ready to go. So now you should be able to see Amass as part of the connectors in here. So that's active, and we're ready to go for this session. And just a couple of words when we're actually inside Claude. There are a few things to consider. If I go to the project here, we can add instructions, we can add context to the project itself. In today's session, I'll actually just use skills. But if you do use context, so let's say if you have your own papers that you're using, then there's also what we call a context window for how much of that these models can actually take into account in terms of let's call it memory at once. For Opus 5 and Sonnet 5 on the paid plans, it's 1 million tokens, and other models can be lower. So this would correspond to roughly 50 to 70 papers. So if you're uploading PDFs of 20 to 30 pages, then this would be supported. And beyond that, what we call a RAG would be used, which is a system for then storing these files in kind of a bulk storage and then giving instructions on where to and how to find the data, which behaves slightly differently and than it would if it's just a smaller data set that you're searching across. And one thing to be aware of, even if we've connected Amass, if we upload a set of papers here, they're not connected in the same way that they would be in Amass. So for instance, when you have an identifier, let's say a molecule that references to clinical trial data that also carries references to literature, it's not that kind of built-in connection, as we would call it, maybe in Amass, where you can actually directly traverse the different nodes from one to another. But Claude is, of course, able to reason then across finding this identifier here and so on. But of course, we all know that there are synonyms as well used for maybe the same molecule, same drug with the marketed name, with the INN, with other program names as well. So it can be quite complex also to find your way through this accurately, at least. All right, so if we start with the first use case, and this will be an asset evaluation. So just going through some evidence from clinical trials and literature related to an asset. And you can imagine that you're working at a biopharma company with deep expertise in maybe a specific target or indication, and a new asset has just come across your desk. This might fit your company strategy. So whether you're then a research scientist digging into the underlying biology, you may be a clinical development lead scoping out the trial or regulatory path, or the BD or market research analyst looking at the commercial opportunity. You're all trying to answer somewhat the same question. So is this a reasonable asset? Does it hold up? And where does it potentially fit? So assuming that you have Claude in your company, the problem now is then that Claude, it can produce reports that are decent. It can create some outputs, but it may not live up to the rigor required for making these kinds of decisions, these high-stakes decisions. And you may also see yourself spending more time quality controlling the outputs and verifying the accuracy than it would otherwise take to maybe Amass that work together. So ideally, maybe in these cases, you want to evaluate the target or mechanism of action, the molecular profile, how it differs from other molecules, what clinical evidence is there for this molecule or this class, and what are the regulatory precedents? Does it have competition? What the IP landscape looks like? So looking at the competitive landscape and patents. And then, of course, combining all these factors. So how differentiated is the asset, what the strength of evidence is for this, what the competitive risk is. And of course, you want to be able to trace everything back to the right PMIDs, NCT numbers, patent numbers, and so on. So for the example itself, I chose an antibody that basically did exactly what it was designed to do. And it's, well, this is going to give it away, but it cleared amyloid plaques from the brain as it was supposed to, confirmed by PET scans. And it did it at a rate where its own maker's model predicted it almost perfectly, but the trials still failed. And the asset in question is Gantenerumab. So it's a biologic asset for Alzheimer's disease, targeting the amyloid beta precursor protein. So basically, this went through around a decade of trials, and it's marketed by Roche, or not marketed actually. So it failed the phase 3 trials in 2022. After a decade-plus long program, there were a number of different programs with the molecule, different trials, and ultimately it did not pass through or pass past phase 3. It's a very it has a very detailed public record and documented pattern. There's also a lot of evidence for anti-amyloid antibodies. And yeah, this was the fourth major anti-amyloid antibody to fail a pivotal trial in the last 10 years. Okay, so how should we then start evaluating this in mass? So of course we could start by gathering some data, gathering some information, asking some questions about how to go about this. So maybe if we didn't have a better approach, we would want to evaluate Gantenerumab as a biologics asset for Alzheimer's disease. Is it worth continued investment? Of course, we know that it failed phase 3 or partnership interest. So the likely answer is no. But is there maybe anything else that we could find from this evidence? So we can go ahead and ask this. And as you can see, well, now Claude is working on it. Unless we specify which assets Claude is using. So in this case, I didn't mention anything about Amass. I didn't have any other connectors available. So we, first of all, maybe want to be able to verify that Claude is using the right tools. And okay, so now Claude already has some context. You can see that it's found some assessment that I previously run. And you can see that it also used a skill here, clinical assessment. So it actually already has some context. So unless it were for this, Claude would actually just do web searches. So try to find this information online. Now with Amass, we're actually working with a skill already. And this is important because this also allows us to get a complete package from the get-go with minimal effort, include the right parts, have some evaluations as well for Claude itself to quality control its work, and provide the format that we want as well in the end. So of course we could specify this in a prompt, but if it's in skill, this will make our lives a whole lot easier. So we could choose to do a full package, report and dashboard only, written analysis only. Maybe we'll just do a report and dashboard for this part. Okay, who is this for? Is it for investment committee, BD licensing evaluation, general due diligence? Let's just go with the investment committee in this case, as an example. So we can just let Claude work through this now. And if we were to maybe specify already from the get-go a skill that we wanted to use for Claude, we can use the / commands to do that. So you can see that I've added a couple of skills here already in advance. And we actually have skills already for Amass available on our website. And it's something that you can also build by yourself in Claude. And it's fairly straightforward. I'll get back to that towards the end of the presentation as well, just to show you how that can be done as well. But if I'll just show you first where you can actually find these skills. I'll just jump over to the Amass website here. And under resources, you can find skills and prompts. And so it's amass.tech, the URL, and then resources, skills and prompts. So scrolling down, you can find a few examples that we've put together for just getting started. So you can filter by skill here if you want. So we have the target prioritization matrix by Medical Scout, trial evidence trace, and other alternatives that we can either directly implement or build on top of. So let's say if we wanted to use trial evidence trace, I'll just click here, and then I can click on download skill. Of course, you can also see down here we have some examples on how to use it. And if I click on download skill, you can see it open here. Then I can just click on Command S and save this. So then we have our skill downloaded, and then I can basically upload this to Claude and start using that already if I want to. So if I just want to do that right now, I can basically, well, I can go to settings, skills, and add a skill here. So I can upload a skill directly. And I have that on my desktop, so I can just grab that here. Great. So let's try uploading this here. Okay, so I already have one here, so I can either upload it and replace, or then just keep the existing one here. I'll just keep the existing one, but this will basically allow you to upload it here. And the trial evidence trace looks like this. And here you can see what's actually outlined in here. So in the trial evidence trace, we are expecting single input for a clinical trial ID, so an NCT number, for instance. And this then reads that specific trial from Amass's trial core. It walks to the PubMed core then, and starts to look for publications that describe this. And this is then how you can look for evidence for a given trial and a drug in a trial. So you can ask, for instance, which papers describe this specific asset, and potentially perform literature review like this. But this can also traverse multiple different cores. So this is a one jump to one of the adjacent cores here. So from the trial core to then PubMed core. But of course, you could jump to DrugCore, to GeneCore, and so on as well, to perform maybe more complex analysis. And I'll actually get back to describing some of the content of the skills right now. But first, we can look at what Claude is actually doing here. So you can see that Claude is leaving a trace of what it's actually doing. It's reading the research plan. It's reading how to use Amass, and then running through different steps as defined in the skill itself. So you can see that we're going through different records in DrugCore, different records in GeneCore. So here you can see the amyloid precursor protein query of that, looking up these in DrugCore by Campbell ID or Ensemble Gene ID, and running through different steps here. Right now we're running multiple different agents at the same time in order to then get to the outputs that we want to see. So of course, if you run a skill like this, it may take a while for Claude to run it. So I've prepared some examples already where we've run this in the interest of time, but we can of course come back to this and look at the output here. But basically, this is how you can already get started using Claude and Amass together to get some of these outputs, which can be very valuable. So I have my pre-webinar workspace here. I've run a couple of different examples. So I also use the skill here for clinical evaluation. And what this basically does is it does a PICO analysis, and also then the pipeline analysis for that as well. And for the different questions that we might be asking, that I mentioned before around validating the target and mechanism of action, in the first place, there's a pretty comprehensive research body around the amyloid beta precursor protein already. Molecular profile, clinical evidence, regulatory precedents, as well as then the competitive landscape. These are then all outputs that we expect to have. But now in Claude, what I also wanted to mention, so one of the outputs here is a briefing, which basically describes the asset itself, so Gantenerumab, and what happened in the trial. So of course, we're looking at a fully human IgG1 monoclonal antibody, as mentioned, targeting the APP. Here you can see that the DrugCore carries 18 linked trials and 0 linked regulatory authorisations, as we'd expect, since it never passed phase 3. And Roche never filed BLA or MAA for it. And you can then jump over to the results, why it failed the trials, and where it got terminated. And also then, if there are some other research areas or other molecules, successors in this landscape. So you can actually see that Roche's chosen successor is trontinemab, and this is fused to an antitransferrin receptor for greater CNS exposure, which is now enrolling in phase 3 trials. And so we can see that actually, in this case, delivery might have solved these issues. And if we look at the output, so the PICO framework, which populations were participated in the programs, the intervention method, comparator outcomes. And then for the pipeline overview, this is a very simple example of what the pipeline can look like here. So we have just a table showing the trial itself, the NCT registry ID, which is pretty important as well to make sure that we actually have the correct ones, which phase the population is, what the trial core gives as a result. So some of these were completed, some of those were terminated, some are potentially then open, what the enrollment looks like, and then the key results from the different trials. So in the output of this report, we also output the successor asset, so potential in developing similar molecules. You can see that we list a chemical ID here as well. So if we wanted to, we can also dig into this and then potentially query more about the different successors. So maybe tell me more about trontinemab. And of course, usually when we prompt, we want to be a bit more specific. This was a very generic one, so Claude may just look up everything here and give you a rewording of what's here. But you can actually see that, okay, so great, it's searching PubMed core, it is searching patent core as well, and now it's getting it by the exact ID that we're referencing, so we can make sure that we're actually getting the exact corresponding data from across these different cores. So now Claude is giving us an answer. It's a successor for Gantenerumab. It's a bispecific fusion, as was mentioned in the report as well. And okay, perfect. Here we have the founding preclinical paper from 2023 in MABS. And that was, where did I lose it? There we go. That was a delivery of the brain shuttle amyloid beta antibody fusion trontinemab. And as Claude says, this is the direct source for the mechanism and the numbers that then are showed or are displayed as results in the preclinical paper. So 4 to 18 times greater brain exposure than Gantenerumab that failed. You can see that there are 4 linked trials, and as for IP, there is a plausible lineage of patents. So there was not a direct one, but there is a one that it may be. So if we're then to research more into therapeutic fusion protein, you can often find these slightly more vague names in patents, and this may then be something to look into as well, right? So then for the competitive context, trontinemab, that's not the only molecule in this space, in the TFR1 brain shuttle bispecific space. There's Janssen, there are other competitors as well. Now, our Claude is still working on it, working on our question here, so I can just let it continue, work through it, but you can see that we're also running a comprehensive search here. Also, here Claude is doing some web searches itself, so getting potential relevant information. It's something that Amass can also include. And now we're generating some results here. But just to illustrate some other results that we may have Claude produce as outputs, it can also be things like interactive graphs. So let's say if we wanted to create something that's more maybe tangible or more easily something that you can visualize more easily. It could also be a graph like this. So we have Gantenerumab. Now we're listing the Amass Cores here as well, and this was an artifact generated by Claude. Of course, there are other ways as well to also display these kinds of visuals if you're using potentially other tools. But this is just to also showcase that each of these nodes here actually also correspond to the actual clinical trial. So this is what Amass also provides, right? So you'll have the hyperlinks to the actual studies themselves. So it's not just a graph that has an NCT number. It's something that you can actually click on and verify as well. So this is also very much what you want in when performing this kind of work. Great. So while Claude is multitasking here still, we can also multitask and jump to the topic of how to create these kinds of skills in the first place. I already showed you how you can kickstart this by downloading an asset from the Amass platform or from our website. But if we jump into here, so again, this was to just illustrate that if we're running Claude, this was me running Claude yesterday without any connectors. It's just basically doing web searches across the web. You could connect it to maybe PubMed or similar databases. But again, it's connecting these individual databases, potentially not as comprehensive as Amass, and you will not be able to obtain those cross-links across the databases. Whereas if you use the Amass connector, is using the tool directly, you can search across all the databases and then get a coherent report or coherent spreadsheet visualization as the output. And also for the outputs for the second example that I had prepared, I don't think we have time to go through the full example here now, but just to showcase what this could look like if we wanted to then look at or perform some kind of other analysis. So it could be a target or a drug class landscape analysis or a report. We could just create that as well. So maybe it could be along the lines of find and let's say prioritize drug targets in the GLP-1 or in creatine signaling pathway for let's do obesity. it's hard to type. Okay, so if we just type in this, Claude would maybe try to find the right instructions to work with. We already have some skills, so I can just add using, and now we can then actually do a clinical pipeline landscape or a drug class landscape. So depending on the actual use case that we have, we can then choose different ones and give Claude instructions right away on how it should actually go about this. So let's just let Claude run this. And I think this will also take a while, but Claude, I think, already will look through some assets that we may already have from before. And okay, great. We have the input, we have the skill that's being used. I defined it here as well with the / command. And okay, let's see. It's built to map an existing drug class. What you described is different. Finding and scoring candidate targets. Okay, so instead, I should actually use this. So here, Claude actually will look up the description of the skill and propose something better. So instead of me doing a futile drug class landscape, I'll rather do a target prioritization matrix here. So that's why also descriptions in some of the skills become important, just to make sure that you're not using the wrong set of instructions, the wrong protocol to analyze the wrong thing. So let's just then go with the target prioritization, and glad that Claude caught this mistake, right? So if we then wanted to look at some of these outputs, I've also prepared some in advance. Okay, so now we're asking, or Claude is asking more questions. We can specify, and these are all something that the instructions actually surface. And this is something that is then very useful for making a skill as broad or as narrow as you'd like. So make sure that Claude asks the right questions in order to also direct the user, maybe you create a skill that a colleague also uses, to direct that colleague into using that skill the right way. So if we were then to look at another example that I run here, this is a cardiovascular risk reduction for the obesity and obese populations analysis. So we can actually then also just take a look at some of the outputs that we have here. So this is shaped as a report, has a wait list, a watch list, so other assets in the same space, threats and regulatory risks, structural risks, and also then other, so the reality check, okay, if we have actual unison in the data set, so does the data match what the registry says, making sure that that we have the right data sets or the correct data that we're working with. And here we're actually listing the different assets here. It's a long list. So it's also a long report. You have the landscape here outlined by phase. This is also something that we can obtain as an interactive report, but this is also something that's very, very useful along then with the links that takes us to these either molecules or other, let's say, specific trials, for instance, that are ongoing. So I know we're approaching time, so I just wanted to wrap up with the last section here, which is the replicable workflows. I already touched on this and we mentioned the skills, but when we're building the skills in general, we just want to be able to approach it from, if we approach it from scratch, what does the skill do? Who should, when should it be used? And then what are the key capabilities that should be defined in the skill? So maybe what are the tools or what are the outputs that Claude should provide? And this then starts from drafting. And when you're using a skill, you want to combine a few different tasks that you expect Claude to do. So starting from 2 to 3 maybe, and explaining which sources should be considered, especially maybe when it comes to Amass, for instance, or any other use case as well. Testing, so having test cases where you can actually verify the actual output. So something that you're familiar with, something where you can spend little time checking that the output is as expected. Also then for the third part, the evaluation part, making sure that when we're using the right sources, that everything is then traceable and can link back to the source. And also that if there's uncertainty, that should be visible in the report. So if some record doesn't have a value, then it should state that. And we should also have the skill include that if we don't find a result, we should state that there's no result for this, or the value was 0 or null, rather than just omitting it or assuming something else, or letting Claude assume something else. And then finally refining this, and basically, as Claude did before, it actually captured that our intent was to use the target prioritization matrix skill rather than the other one, which is the incorrect one to use for our query. So that can also capture or make sure that the Claude is or you're using the right skills for the right use case. And you can build these in Claude as well. You have the skill builder skill in Claude, so you can also start from scratch. But as I mentioned, you could also just take an Amass skill and bring that into Claude and then edit that as you'd like. And I know we're a couple of minutes over time, so I'll just wrap up with this. So if there are any takeaways from this session, one is that we just need to give Claude the right tools to work with. In the instance of Amass, we'll return traceable and reliable results, grounding the analysis. So making sure that we're actually using the right search tools, finding the right results, and then fetching the right links and identifiers for you to work with in the first place. And using skills as a framework for performing the work, this is also important for actually having, like any protocol in a lab, for instance, you need to make sure that you're doing the same things in the same way, have clear instructions for the user, or in this case, Claude, to perform the different steps using the right tools, having the right context, and also maybe verification steps involved. And of course, as with anything, it's not, so LLMs, they're not deterministic, as you would maybe get the same results when you run a script in Python. You will not get the same results when having, or while having the same result, the same prompt for Claude each time. So making sure that you also refine and iterate, whether it's a skill or whether it's how to come to a result, and then building that into a skill, and providing the right context as well to not skew the analysis in one way or the other. That's also quite predominant, or yeah, it's very important as well to make sure that this is something that you think about. Okay, so I know our Claude is still probably running in the background. I was asking for a question here. Thank you, Camila, for pointing that out. For the first query, this is quite a long skill that we're running, so you can see that we're still, Claude is still assessing it. So we'll get a pretty detailed report, similar to what I showed you here. So I'll let Claude continue working in the background and wrap up this session. If you have any questions, you can, of course, just reach out directly to me, and then I'll be happy to have a call with you as well, or exchange messages over email. But I want to thank you for your attention, and now we can move on to the Q&A. So if you have any questions, you can just start asking them right now, and I'll be happy to answer them. And if you have any any questions later on over email? I can also just share my email address here. There we go. Any questions from the audience? We have a question. Sorry, maybe I missed it. Okay, so the question here is, what is the advantage of using Claude together with Amass connectors instead of only Amass? And yes, that's a very good question from Anas. And the answer is, well, there are some trade-offs, right? So as I mentioned before, we focused on on Claude in this webinar, but we also do have an Amass application. And while we don't have time to go into that specific application or use cases, what you can do in the Amass app during this webinar, the differences are maybe, so both are using the same underlying data set, so the same underlying cores as I showed here. So the app, the MCP, are all calling the same data sets from here. The difference is then that in Amass, we're doing that directly. In Claude, you have to maybe explicitly say that this is what you want to do. You want to look it up in Amass. Claude will have maybe specific abilities. Let's say if you build skills in Claude, you can do maybe different things with them versus what you would do in the Amass application, where it's already more predefined. So you have more freedom maybe to do to combine Claude with different things than you may have in the Amass application. Whereas Amass, it's excellent for specific work related to anything that you want to do directly related to this kind of these kinds of data sets and this kind of research. So Amass, the application is very much more specific and gives you direct results from this. You also have some visualizations out of the box there. And in Claude, you have to maybe define these outputs yourself as well a bit better. And yeah, things like inline references, that's something that you can get in the Amass app. Claude doesn't provide that, unfortunately, so you cannot get, let's say, a citation inline in the output directly. Any other questions from the audience? All right, so if nothing else, then I'd like to thank everyone for joining. And as I mentioned, the webinar recording will be accessible after the webinar as well. So if you need to go back to this. Okay, we have another question actually. So let's take a question from Shiksha here. Have you done any benchmarking with others comparing the comprehensiveness of the answers? So for the benchmarking part, I alluded to this in the slides. We've done some benchmarking, and of course it depends on what exactly you want to benchmark. But some examples, one example that we have here is benchmarking that we did for literature or biomedical retrieval. And in this example, we compared the tools, so just using Claude with the PubMed connector and then using Amass. And of course, these are very complicated things to evaluate and benchmark. But what we found was, so looking at what is actually retrieved is the comprehensiveness of the retrieval of the articles retrieved. Does it cover the extent of the question? And then the precision of these. So of these papers that were retrieved, were they relevant? And for this, we found that only Claude using the PubMed connector performed worse than using Amass, either the application or Claude with the Amass connector. So this is the kind of benchmarking that we have done, and we may do more in the future. But these are this is one concrete example where we've then shown this in a more structured way and done our own benchmarking for this. Great. Any other last questions before we wrap up? So we're almost about to. So great. Happy to have had everyone join for today. And yeah, again, if you have questions, You can see my email on the screen, or just reach out to us through the website and ask any questions if you think about anything afterwards as well. So yeah, thanks for today, and have a good rest of the day.